Hello, dear friends, and welcome. I'm so happy to have you here. As we begin, I'd love it if you could share in the comments where you're joining from and the time of day it is for you right now. It's always wonderful to witness people tuning in from across the world. Thank you as well to those who have already subscribed and shared their support by liking the videos. It helps more than you know. And please don't hesitate to leave your ideas or requests for future videos. Let's get started. Chapter 1: Understanding Stem Cells and Their Types. Stem cells are remarkable entities within our bodies, the building blocks of life that possess a unique ability to develop into many different cell types. This extraordinary characteristic allows them to play crucial roles in growth, healing, and regeneration. Imagine, if you will, a small group of cells that can transform into heart cells, skin cells, or even neurons. This potential is what makes stem cells so captivating to scientists and medical researchers alike. At the core of their definition, stem cells are distinguished by two primary properties: self-renewal and differentiation. Self-renewal is the process through which stem cells can divide and produce more stem cells, maintaining their population over time. Differentiation, on the other hand, refers to their capacity to develop into specialized cells. This duality forms the foundation of their importance in biology and medicine. There are two main categories of stem cells: embryonic and adult stem cells. Embryonic stem cells, derived from the early stages of an embryo, are characterized by their pluripotency. This means they can give rise to any cell type in the body. In contrast, adult stem cells, often found in various tissues such as bone marrow or fat, are typically multipotent. This limits their ability to differentiate into a narrower range of cell types compared to their embryonic counterparts. To visualize pluripotency, consider a vast library filled with countless books. Each book represents a different cell type, waiting to be chosen. Pluripotent stem cells, like the newly opened library, have access to all the knowledge contained within. Multipotent stem cells, however, are more like a specific section of that library, perhaps only containing books on a particular subject, such as the medical sciences. While their range is narrower, they are nonetheless invaluable for tissue repair and regeneration. The potential of both types of stem cells is profound, especially in the context of development and repair. During the early stages of embryonic development, pluripotent stem cells contribute to the formation of all the different tissues and organs that will ultimately make up a fully functioning organism. Each division and differentiation of these cells is a carefully orchestrated process, guided by intricate signaling pathways and genetic instructions. This dynamic process illustrates the beauty of life unfolding, cell by cell, as the embryo grows and develops. As development progresses, adult stem cells take on their vital roles. They reside within tissues, ready to spring into action when needed. For example, when a bone is fractured, the adult stem cells in the bone marrow activate to produce new bone cells, facilitating the healing process. This remarkable ability of adult stem cells to respond to injury and repair damaged tissues is a testament to their importance in maintaining our body's health and functionality. The significance of stem cells in research and medicine cannot be overstated. They hold immense promise for regenerative therapies, potentially allowing us to treat conditions that currently have no cure. The work of pioneering scientists like Shinya Yamanaka and James Thomson, who were instrumental in uncovering the potential of stem cells, has opened new avenues for understanding and harnessing these remarkable cells. Yamanaka famously stated, "The future is not about finding new stem cells, but about understanding how to use them." Imagine the possibilities if we could harness the power of stem cells to regenerate damaged tissues, restore lost functionalities, or perhaps even reverse the effects of aging. These questions drive researchers to explore the boundaries of what stem cells can achieve. The potential applications span a wide range, from treating degenerative diseases to improving recovery from injuries. The exploration of stem cells raises fascinating ethical questions, particularly when it comes to embryonic stem cells. The debate surrounding their use often centers on the moral implications of using human embryos for research. This dialogue is crucial, as it shapes the direction of scientific inquiry and the regulatory frameworks that govern stem cell research. Striking a balance between scientific advancement and ethical considerations is a challenge that continues to evolve. As we contemplate the future of stem cell research, it is essential to recognize the journey that led us here. The early discoveries that laid the groundwork for our understanding of these cells were often met with skepticism. Yet, through dedication and perseverance, scientists have gradually unraveled the mysteries of stem cells, illuminating their potential and promise. The landscape of stem cell research is constantly changing, with new breakthroughs emerging regularly. Scientists are refining techniques to manipulate stem cells, enhancing their ability to differentiate into specific cell types. Advances in technology, such as CRISPR gene editing, allow for precise modifications at the genetic level, paving the way for innovative therapies that could one day change the course of diseases. The elegance of stem cell research lies not only in its scientific complexity, but also in its profound implications for humanity. Imagine a world where spinal cord injuries could be treated with personalized cell therapies, or where degenerative diseases such as Parkinson's could be managed or even reversed. The vision of harnessing stem cells to improve quality of life and heal the body is both inspiring and motivating. As we journey deeper into the world of stem cells, let us appreciate the intricate balance between hope and caution that guides this field. Each discovery brings us closer to understanding these remarkable cells and their potential. The stories of dedicated scientists, their triumphs, and their challenges remind us of the human spirit's resilience and curiosity. In this exploration, we are reminded of the interconnectedness of life itself. Every cell in our body has a story to tell, and stem cells sit at the heart of this narrative. They are a bridge between what has been and what could be, embodying the essence of growth, healing, and the endless possibilities that lie ahead. As we continue to uncover the mysteries of stem cells, let us remain open to the wonders of science and the profound impact it can have on our lives. The journey of understanding stem cells is just beginning, and the horizon is filled with promise and potential. In the realm of stem cell biology, the distinctions between pluripotent and multipotent stem cells extend beyond their sources and capabilities. They also illuminate the intricate mechanisms that govern cellular fate. Pluripotent stem cells, as we have seen, hold the extraordinary ability to transform into any cell type, a characteristic that is not merely a result of their origin, but is intricately linked to the regulatory networks that control their behavior. These networks involve a symphony of genes and signaling molecules, orchestrating the delicate balance between self-renewal and differentiation. Imagine entering a bustling city, where every street and alley is alive with movement and purpose. Each building serves a unique function, much like the various cell types within our bodies. In this vibrant city, pluripotent stem cells act as the architects, possessing the blueprint to design and create any structure needed. Their potential is a reflection of their complex internal programming, which remains a focal point for researchers aiming to unlock the secrets of development. Adult stem cells, on the other hand, serve a more specialized role akin to skilled craftsmen in specific trades. These multipotent cells, found in tissues such as the bone marrow, skin, and intestines, have a more defined path. They are equipped to generate a limited range of cell types necessary for maintaining the health and functionality of their respective tissues. For example, hematopoietic stem cells in the bone marrow are responsible for producing various blood cells, ensuring our bodies are well equipped to transport oxygen and fight infections. This specificity is crucial, allowing adult stem cells to provide timely responses to injury or wear and tear. The beauty of this specialization lies in its efficiency. When a tissue is damaged, adult stem cells swiftly mobilize, activating their regenerative capabilities. Picture a serene garden where each flower represents a different cell type, flourishing in its designated space. Adult stem cells act as the gardeners, tending to the flowers, nurturing them back to health when storms arise. This nurturing role is vital for sustaining life, as it allows our bodies to repair and regenerate, maintaining balance and vitality. The interplay between pluripotent and multipotent stem cells is a fascinating dance of specialization and versatility. As we analyze their roles in development and repair, we find ourselves asking profound questions. What if we could harness the versatility of pluripotent stem cells while also capitalizing on the targeted efficiency of multipotent stem cells? The answer lies in the ongoing research that seeks to bridge these two worlds, exploring the potential of induced pluripotent stem cells. Induced pluripotent stem cells, or iPSCs, are derived from adult cells that have been reprogrammed to an embryonic-like state. This groundbreaking discovery, pioneered by Shinya Yamanaka, revolutionized the field of regenerative medicine. Imagine a skilled artist who can take a canvas of any type and transform it into a masterpiece, regardless of the original image. iPSCs represent this artistic potential within stem cell biology, offering a route toward generating patient-specific cells for therapies. This innovation not only circumvents ethical concerns surrounding embryonic stem cells, but also opens avenues for personalized medicine, where treatments can be tailored to the individual's unique cellular makeup. The implications of manipulating stem cells extend beyond mere regeneration. They touch upon the very essence of what it means to heal. Consider the potential to create neurons from a patient's skin cells to treat neurological disorders. The vision of restoring lost functions or alleviating debilitating conditions is invigorating and represents a frontier that researchers are eagerly exploring. Each breakthrough in this field brings us closer to a reality where the body can heal itself in ways once thought impossible. Yet, the path forward is not without challenges. The intricacies of stem cell behavior are still being unraveled, and researchers must navigate the complexities of cellular environments and the signals that govern stem cell fate. It is a journey marked by both triumphs and setbacks, each contributing to a greater understanding of these remarkable cells. As we reflect on the pioneers who dared to dream beyond the current limits of science, we find inspiration in their resilience and commitment. Throughout history, remarkable figures have shaped our understanding of stem cells. Gurdon and Yamanaka, both awarded the Nobel Prize for their groundbreaking work, have emphasized the significance of curiosity and exploration in scientific endeavors. Gurdon once remarked, "The most important thing is to get young people engaged in science," a sentiment that resonates deeply in the field of stem cell research. The pursuit of knowledge is a collective effort, driven by the questions we ask and the discoveries we make together. As we contemplate the future of stem cell research, we are left with a sense of wonder at the possibilities that lie ahead. Imagine a future where we can not only repair damaged tissues, but also enhance the body's own regenerative capabilities. With ongoing research, scientists are delving into gene therapies that may one day complement stem cell treatments, creating a holistic approach to healing and health. The importance of stem cells in development and repair is a testament to the resilience of life itself. They remind us of the intricate connections that bind us, as every cell in our bodies plays a role in the grand narrative of existence. The journey of understanding stem cells is not just a scientific endeavor. It is a reflection of our innate desire to heal, to grow, and to transcend the limitations of our biology. In this exploration, we may find solace in the knowledge that the mysteries of life are not merely puzzles to be solved, but are also invitations to wonder and reflect. Each discovery in stem cell research beckons us to consider the profound implications for our future, urging us to remain open to the possibilities that await. As we continue to uncover the secrets of these remarkable cells, we step into a realm rich with potential, where the boundaries of science and imagination intertwine. Chapter 2: The Process of Stem Cell Differentiation. Differentiation is a wondrous journey, a metamorphosis that transforms the humble stem cell into a myriad of specialized cell types. This transition is not merely a change in form, but a profound evolution of function. Understanding differentiation is essential, for it is through this process that stem cells fulfill their vital roles in development, healing, and regeneration. Imagine a blank canvas gradually revealing a vibrant landscape, where each stroke of color represents a unique cell type, each playing its part in the symphony of life. The significance of differentiation extends far beyond the individual cell. It is the foundation upon which complex organisms are built. From the earliest days of life, when a single fertilized egg begins to divide, differentiation sets into motion the intricate processes that lead to the formation of tissues and organs. This remarkable orchestration is overseen by a series of stages that stem cells traverse as they embark on their transformative journey. The first stage of differentiation begins with the decision-making process of a stem cell. What will it become? This choice is influenced by intrinsic factors, such as the genes that are activated or silenced within the cell. Each stem cell carries the potential to develop into various cell types, but it is the activation of specific genes that steers the cell down a particular pathway. This stage is akin to standing at a crossroads, where the path taken will lead to vastly different destinations. As the stem cell progresses, it enters the second stage, characterized by a gradual commitment to a specific lineage. During this phase, the stem cell begins to express markers that are characteristic of its destined cell type. For example, a stem cell on its way to becoming a neuron will start to express genes associated with neuronal function. This commitment is not instantaneous, but rather a gradual shift, much like the changing hues of a sunset as night approaches. The third stage involves further maturation and specialization. Here, the cell refines its identity, acquiring the characteristics and functions that define its role in the body. This stage is where the magic truly happens. The stem cell transforms into a fully functional cell, displaying the unique features that allow it to perform its designated tasks. Imagine the intricate process of a caterpillar metamorphosing into a butterfly, emerging with beauty and purpose after a period of change. However, the journey of differentiation is not solely dictated by the internal workings of the stem cell. External factors play a pivotal role in guiding this process. Various signals from the surrounding environment can influence how a stem cell differentiates. These factors include growth factors, hormones, and even physical cues from neighboring cells. This interplay between the internal and external is a delicate dance, ensuring that stem cells respond appropriately to the needs of the organism. Microenvironments, often referred to as niches, serve as the context in which differentiation occurs. These niches are specialized environments that provide the necessary signals for stem cells to thrive and differentiate. They can be found in various tissues throughout the body, such as bone marrow, where hematopoietic stem cells reside, or in the brain, where neural stem cells are nurtured. Each niche is uniquely tailored to support specific types of stem cells, fostering their growth and guiding their fate. The role of microenvironments becomes even more fascinating when we consider the concept of stem cell plasticity. Imagine a stem cell that can adapt to different environments, responding to changes in its surroundings. This flexibility is crucial as it allows stem cells to adjust their differentiation pathways based on the needs of the organism. It raises the question, how do these cells sense their environment so acutely? The answer lies in their ability to receive and interpret a myriad of signals. One of the most remarkable aspects of differentiation is its potential for therapeutic applications. Scientists are exploring how to harness the power of stem cells to regenerate damaged tissues and treat various diseases. The concept of using stem cells to rebuild an injured heart or to create new neurons in the brain is no longer relegated to the realm of science fiction. As we learn more about the processes that guide differentiation, the possibilities for treatment expand. Consider the journey of an extraordinary scientist, Shinya Yamanaka, who was awarded the Nobel Prize for his groundbreaking work on induced pluripotent stem cells. His discovery that adult cells could be reprogrammed to behave like embryonic stem cells opened new avenues for research and therapy. Yamanaka's work exemplifies the potential for differentiation to be manipulated for the benefit of humanity, allowing us to reimagine the possibilities of healing and regeneration. As we reflect on the intricacies of stem cell differentiation, we are reminded of the remarkable adaptability of life. Each cell, in its journey from a stem cell to its specialized form, embodies a story of growth, change, and purpose. The stages of differentiation unfold like the petals of a flower, revealing the beauty and complexity of the biological world around us. What might the future hold as we continue to unravel the mysteries of stem cell differentiation? Will we unlock the secrets to growing entire organs in the lab, or perhaps develop therapies that can reverse the effects of aging? The potential is as vast as the universe itself, limited only by our imagination and our commitment to understanding the science that underpins life. In contemplating the role of microenvironments, we begin to appreciate the exquisite nature of cellular interactions. Each stem cell exists not in isolation, but as part of a community, influenced by its surroundings. The dynamic interplay between stem cells and their niches highlights the importance of context in the differentiation process. It leads us to ponder, how can we create optimal conditions for stem cells to thrive and fulfill their potential? As we journey through the landscape of stem cell biology, we encounter a world where the boundaries of possibility are constantly expanding. The understanding of differentiation is not merely an academic pursuit. It is a quest to harness the power of life itself. Each discovery brings us closer to unlocking the potential of stem cells for regenerative medicine, illuminating pathways that were once shrouded in mystery. The process of stem cell differentiation is a testament to the beauty of nature's design. It is a symphony of signals, a choreography of genes and environments, all working in harmony to create the diverse tapestry of life. As we continue to explore this fascinating realm, we stand on the precipice of exciting discoveries, eager to learn how we can guide the dance of differentiation and harness its power for the betterment of humanity. As we traverse the complex pathways of stem cell differentiation, we arrive at an understanding of the various factors that influence these pathways. This journey is not only a biological process, but also a dance of molecular interactions that orchestrate the fate of each stem cell. Imagine a grand conductor leading an orchestra, where each section must harmonize to produce a beautiful symphony. The conductor in this case is a myriad of external signals that guide stem cells through their developmental journey. Among the most significant influencers are growth factors, which are proteins that stimulate cellular growth, proliferation, and differentiation. These factors bind to specific receptors on the surface of stem cells, initiating a cascade of intracellular events that ultimately influence gene expression. For instance, fibroblast growth factor plays a critical role in the early stages of stem cell differentiation, particularly in the formation of neural tissue. The binding of this growth factor activates pathways that promote the transition from a stem cell to a neuron. What might happen if these growth factors are absent or dysfunctional? The consequences could be severe, leading to developmental disorders or impaired healing processes. The intricate balance of these signals is essential for maintaining the health and functionality of stem cells. The importance of this balance invites us to ponder: how can we manipulate these signals to enhance regenerative capabilities? Hormones also play a pivotal role in this delicate interplay. They circulate throughout the body, delivering messages that influence stem cell behavior. For example, insulin-like growth factor is known to promote the proliferation of mesenchymal stem cells and their differentiation into various cell types, including bone and cartilage. Imagine a steady stream of nourishment flowing toward a plant, encouraging it to grow strong and resilient. In a similar vein, hormones provide the necessary encouragement for stem cells to flourish and commit to their destined paths. Another fascinating aspect of stem cell differentiation is the influence of physical cues from the microenvironment. These cues can include the stiffness of the substrate on which stem cells reside or the presence of extracellular matrix components. Research has shown that stem cells are highly sensitive to the mechanical properties of their surroundings. For instance, stem cells cultured on softer matrices are more likely to become neurons, while those on stiffer surfaces tend to differentiate into bone cells. This responsiveness to physical context raises intriguing questions. To what extent can we engineer environments that guide stem cells toward desired outcomes? In further exploring the role of microenvironments, we come across the concept of cellular niches. These niches are not merely passive surroundings, they actively regulate stem cell behavior. They provide a sanctuary where stem cells can reside, multiply, and receive the signals necessary for differentiation. Different niches cater to different types of stem cells, each meticulously designed to meet the unique needs of that cell type. For instance, the niche of hematopoietic stem cells in the bone marrow is rich in specialized cells, signaling molecules, and extracellular matrix components that support the maintenance and differentiation of blood cells. The complexity of these niches becomes even more evident as we consider the interaction between different cell types within them. Stem cells do not exist in isolation. They communicate with neighboring cells, which can further influence their differentiation. This cellular communication is facilitated by direct contact or through the secretion of signaling molecules. The orchestrated interplay of signals and interactions within the niche exemplifies the intricate web of life. One might wonder, how can we leverage this knowledge to enhance regenerative therapies? As we contemplate the future of stem cell research, we are reminded of the visionary scientists who have shaped this field. Take, for instance, the work of James Thomson, who was the first to derive human embryonic stem cells in a laboratory setting. His pioneering efforts opened the door to a new realm of possibilities, allowing researchers to explore the potential of these cells in regenerative medicine. Thomson's contributions underscore the importance of understanding not just the science of stem cells, but also the ethical implications and responsibilities that come with such powerful tools. The ongoing exploration of stem cell differentiation invites us to consider the ethical landscape surrounding this research. As we unlock the secrets of how stem cells can transform and heal, we must also navigate the moral complexities of such power. The questions are profound. What responsibilities do we hold in harnessing this potential? How do we ensure that advancements in stem cell therapies are accessible and equitable? As we reflect on these themes, the notion of differentiation morphs from a purely biological process into a deeply philosophical inquiry. The journey of a stem cell, shaped by internal programming and external influences, mirrors our own paths in life, where choices and circumstances converge to define who we become. In the realm of stem cells, the potential for innovation and healing is immense. As researchers delve deeper into the mechanisms of differentiation, the horizons of possibility expand. Each discovery brings us closer to understanding how we can harness these cells to regenerate tissues, reverse diseases, and perhaps even extend the human lifespan. With every revelation comes a renewed sense of wonder about the intricacies of life and the potential that lies within each cell. The journey through stem cell differentiation is not just an exploration of biological processes. It is a celebration of life itself, a testament to the resilience and adaptability of living organisms. As we remain curious about the future of stem cell science, we are reminded that we are but explorers on the frontier of understanding. The path ahead is filled with questions waiting to be answered, mysteries yearning to be unraveled, and discoveries that could reshape our understanding of health and disease. What doors will we open as we continue to learn more about the transformative power of stem cells and their differentiation? Only time will tell, but the journey promises to be as enlightening as it is inspiring. Chapter 3: Molecular Mechanisms Driving Differentiation. In the vast landscape of cellular biology, the journey of differentiation is guided by intricate signaling pathways that play crucial roles in determining cell fate. These pathways act as the communication systems within the cell, orchestrating the complex symphony of events that lead to specialization. Imagine a grand conductor waving a baton over an orchestra, ensuring that each musician knows when to play their part. Similarly, signaling pathways facilitate the exchange of information between cells, influencing their behavior and guiding them toward their destined roles. One of the key players in this orchestration is the notch signaling pathway. This pathway is particularly fascinating due to its role in determining cell fate through direct cell-to-cell communication. When one cell expresses a specific ligand, it binds to the notch receptor on an adjacent cell, triggering a cascade of events that ultimately influences the fate of that neighboring cell. In this manner, notch signaling can suppress the differentiation of neighboring stem cells, allowing for the maintenance of a reservoir of undifferentiated cells. This process is crucial in developing tissues where a delicate balance of different cell types is necessary for proper function. Another prominent pathway involved in differentiation is the WNT signaling pathway, which is essential for the regulation of stem cell proliferation and differentiation. Picture a pathway that guides cells through a dense forest, illuminating their way to specific destinations. WNT signaling initiates a series of interactions that lead to the activation of target genes, crucial for the development of various tissues. When WNT signals are present, they can promote the formation of different cell types, including neurons and muscle cells. The intricate balance of WNT signaling, therefore, is vital for proper development, highlighting how the absence or dysregulation of these signals can lead to disorders. Additionally, the hedgehog signaling pathway plays an equally significant role in differentiation, particularly during embryonic development. This pathway gets its name from the striking resemblance of mutant fruit flies with disrupted hedgehog signaling to the spiky creature after which it's named. Hedgehog signaling is crucial for the patterning of various structures in developing embryos, including limbs and the central nervous system. The activation of this pathway helps in establishing the anterior-posterior axis, guiding cells toward their specific functions. As we explore these pathways, it becomes clear that they do more than just direct cellular actions. They also play a vital role in regulating gene expression. Gene expression is the process through which information encoded in genes is transformed into functional products, such as proteins that perform cellular functions. The regulation of this process is fundamental in determining how and when a stem cell differentiates into a specialized cell type. Without precise control over gene expression, the intricate dance of differentiation would falter. Transcription factors serve as the master regulators of gene expression, acting like skilled artisans who shape the final outcome. These proteins bind to specific DNA sequences, either promoting or repressing the transcription of target genes. Each transcription factor can be thought of as a key, unlocking the door to a specific set of genes that determines cell identity. For instance, the transcription factor OCT4 is crucial for maintaining pluripotency in embryonic stem cells. When OCT4 is present, it allows stem cells to remain in their undifferentiated state, ready to transform into various cell types. However, as differentiation begins, the levels of OCT4 diminish, giving way to other transcription factors that drive the process towards specific cell types. Another pivotal transcription factor is SOX2, which works alongside OCT4 to maintain the pluripotent state of stem cells. Imagine these transcription factors as companions on a journey, guiding the stem cells along a path that can lead to numerous destinations. The interplay between OCT4, SOX2, and other factors like nano-G creates a network of regulation that ensures stem cells remain flexible and capable of differentiation when necessary. As differentiation progresses, the role of transcription factors shifts. New factors come into play, dictating the fate of the cell. For example, during the development of neurons, the transcription factor neurogenin is activated, promoting the expression of genes necessary for neuronal identity. This activation marks a significant transition from a pluripotent state to a specialized one, demonstrating how transcription factors can direct the destiny of a cell. While signaling pathways and transcription factors are crucial, the story of differentiation does not end there. Epigenetic modifications add another layer of complexity to stem cell behavior, influencing how genes are expressed without altering the underlying DNA sequence. Imagine a library where each book represents a different cell type. Epigenetic modifications serve as bookmarks, determining which books are accessible and which remain closed. These modifications can include the addition of chemical groups to DNA or histones, the proteins around which DNA is wrapped. One notable epigenetic modification is DNA methylation, which generally represses gene expression when methyl groups are added to specific sites on the DNA. This process is critical during differentiation, as it helps silence genes associated with pluripotency and activates those required for specialized functions. Another vital mechanism is histone modification, where the tails of histone proteins are altered, leading to changes in chromatin structure. This alteration can either promote or inhibit the accessibility of DNA, ultimately influencing which genes are expressed. The interplay between these epigenetic modifications and transcription factors creates a dynamic environment, allowing stem cells to respond to their surroundings. Environmental signals, such as those from neighboring cells or the extracellular matrix, can trigger epigenetic changes that guide differentiation. This responsiveness is essential for the proper development of tissues, ensuring that cells can adapt to their context and perform their designated roles effectively. As we consider the remarkable world of molecular mechanisms driving differentiation, it becomes apparent how tightly woven these processes are. The delicate balance of signaling pathways, the precise regulation of gene expression through transcription factors, and the subtle influences of epigenetic modifications all contribute to the orchestration of cell fate. Each component plays a vital role in ensuring that stem cells can navigate their path to specialization with grace and precision. The wonder of this system lies not just in its complexity, but also in its elegance. It is a testament to the beauty of life at the molecular level, where tiny interactions and modifications can lead to the creation of the intricate tapestry of specialized cells that form our bodies. As we marvel at these processes, we may ponder the potential for harnessing this knowledge. How might we influence these mechanisms to repair damaged tissues or treat diseases? The answers lie in continuing to explore the fascinating interplay of molecular forces that shape our very existence. The importance of gene expression regulation cannot be overstated in the context of differentiation. It acts as the cornerstone of cellular identity, dictating which proteins are produced and, consequently, how a cell behaves. Imagine a finely tuned instrument where each note signifies a different gene being expressed. The precise regulation of these genes ensures that the correct proteins are synthesized at the right time, enabling a cell to fulfill its designated role within the organism. Central to this regulation is the role of transcription factors, which are proteins that bind to specific regions of DNA and modulate the transcription of target genes. Think of these factors as skilled artisans, each with a unique craft, shaping the final outcome of the cell's development. They can act as activators, increasing the likelihood of transcription, or as repressors, decreasing it. This dual ability allows them to finely tune the gene expression landscape, adapting to the needs of the cell as it differentiates. One particularly noteworthy transcription factor is C/Abpalfa, which plays a pivotal role in the differentiation of various cell types, including adipocytes or fat cells. C/Abpalfa serves as a master regulator that initiates the cascade of events leading to the formation of adipose tissue. Through its actions, it orchestrates the expression of genes involved in lipid metabolism, ultimately guiding preadipocytes to mature into fully functional fat cells. The presence of this transcription factor not only influences the fate of individual cells, but also contributes to the overall metabolic health of the organism. As we consider the profound impact of transcription factors, it's important to recognize that their influence is not isolated. The cellular environment, including signaling molecules and mechanical forces, plays a vital role in shaping the expression of these factors. For instance, the presence of specific growth factors can activate signaling pathways that lead to the production of transcription factors essential for differentiation. This interplay highlights the complexity of cell fate determination, where external signals and internal regulators converge to define the developmental trajectory of a cell. The dynamic nature of gene expression is further enhanced by epigenetic modifications, which act as regulatory mechanisms that fine-tune transcriptional activity without altering the underlying DNA sequence. These modifications can be likened to a series of switches that can turn genes on or off, thereby influencing a cell's identity. For example, the addition of acetyl groups to histones, proteins that help package DNA, can lead to a more relaxed chromatin structure, allowing transcription machinery easier access to the DNA. Conversely, methylation can lead to a more compact structure, effectively silencing gene expression. Moreover, the impact of epigenetic modifications extends beyond mere gene regulation. They can also influence the stability of a differentiated state. Once a cell has settled into its specialized role, the epigenetic landscape must remain stable to prevent any unwanted reversion to a pluripotent state. This stability is crucial, as it ensures that cells maintain their identity and function within the larger context of tissues and organs. As we delve deeper into the world of epigenetics, we encounter fascinating examples of how these modifications can be influenced by environmental factors. Nutrition, stress, and even exposure to toxins can induce epigenetic changes that affect gene expression patterns. This raises intriguing questions about the long-term implications of our lifestyle choices. How do the decisions we make today resonate through the epigenetic landscape of our cells, potentially influencing not just our health, but that of future generations? A striking illustration of this concept can be found in studies examining the effects of maternal nutrition on offspring. Research has shown that when pregnant animals are subjected to a nutrient-deficient diet, the epigenetic modifications in their offspring can lead to altered metabolic pathways, predisposing them to obesity and diabetes later in life. These findings illuminate the profound interconnectedness of biology and environment, how the experiences of one generation can echo through the molecular mechanisms of the next. In the realm of stem cell biology, the interplay of signaling pathways, transcription factors, and epigenetic modifications forms a cohesive framework that orchestrates differentiation. This intricate dance ensures that cells can respond to their surroundings and adapt to the demands of their environment. Each component contributes to a larger narrative, weaving together the threads of molecular mechanisms that drive differentiation. As we reflect on the elegance of this system, we may find ourselves captivated by the potential it holds. The ability to manipulate these molecular mechanisms opens up exciting avenues for regenerative medicine and therapeutic interventions. Imagine harnessing the power of transcription factors to coax stem cells into specific cell types for tissue repair. Envision the possibilities of using epigenetic therapies to reprogram cells for healing, essentially rewriting the narrative of injury and disease. The journey of differentiation is not merely a scientific exploration, it is a glimpse into the heart of life itself. Each cell, each decision, and each molecular interaction contributes to the beautiful complexity of living organisms. In this vast world of molecular mechanisms, we uncover not only the secrets of development and differentiation, but also the profound potential for healing and renewal. As we continue to explore these themes, we unlock the doors to a future where the mysteries of cellular behavior may one day be harnessed for the betterment of all. Chapter 4: Types of Specialized Cells Formed from Stem Cells. Within the intricate tapestry of life, specialized cells emerge as the unsung heroes of biological function. These cells, arising from the transformed essence of stem cells, serve as the foundation for every tissue and organ in our bodies. Imagine a world where each specialized cell type is a unique instrument in a grand symphony, each contributing its unique sound to create harmony within the organism. Among the many specialized cells that stem cells can produce, neurons stand out as remarkable entities of communication. Neurons are the messengers of the nervous system, transmitting signals throughout the body and facilitating our thoughts, sensations, and movements. Picture, if you will, the delicate structure of a neuron, with its long axons and branching dendrites, resembling a tree reaching out towards the sky. Each neuron is capable of forming intricate networks, allowing us to experience the world and react to our environment. The average human brain contains billions of these cells, all working in concert to process information and sustain consciousness. The pioneering work of scientists like Santiago Ramon y Cajal, often referred to as the father of modern neuroscience, revealed the complexity of these connections, leading to the understanding of how our thoughts and emotions are grounded in cellular activity. Another vital type of specialized cell is the cardiomyocyte, the heart's muscle cell. These cells are responsible for the rhythmic contractions that pump blood throughout the body, ensuring that every organ receives the oxygen and nutrients it needs to function. Imagine the heart as a powerful engine, with cardiomyocytes as its diligent workers, contracting and relaxing in unison to maintain life. The heart's ability to adapt to different demands, such as during exercise or rest, is largely attributed to the unique characteristics of these cells. The work of researchers like Dr. Paul Zimmet has illuminated how understanding cardiomyocyte behavior can pave the way for new treatments for heart diseases, emphasizing the importance of these specialized cells in maintaining our health. Blood cells, too, are a crucial component of our body's functionality. They arise from hematopoietic stem cells located in the bone marrow. These specialized cells come in various forms, each with its specific role. Red blood cells, for instance, are the body's oxygen carriers, resembling tiny discs that glide through blood vessels, delivering life-sustaining oxygen to every corner of the body. White blood cells, on the other hand, are our defenders, equipped to combat infections and protect against disease. Imagine them as vigilant sentinels, scanning for intruders and responding with precision to ensure our well-being. The dynamic nature of blood cells ensures that our immune system functions optimally, reflecting the remarkable diversity that stem cells can create. The functional roles of these differentiated cells cannot be overstated. Each specialized cell type contributes to the overall health and homeostasis of the organism. Neurons, cardiomyocytes, and blood cells work in concert, forming a complex web of interactions that sustain life. For instance, consider how the heart and brain communicate. The brain sends signals to the heart to regulate its pace during moments of stress or relaxation, demonstrating the interconnectedness of specialized cells. This harmony among diverse cell types is essential for the organism's survival, allowing it to adapt to changing environments and maintain balance. The importance of diversity in cell types cannot be overlooked. Each specialized cell type is tailored to perform specific functions, and the presence of a wide array of cells ensures that the body can respond to various challenges. This cellular diversity is akin to a well-stocked toolbox, where each tool serves a unique purpose. Without the full complement of specialized cells, the body would struggle to maintain its intricate balance. The resilience and adaptability of organisms are largely attributed to this diversity, allowing them to thrive in an ever-changing world. As we explore the fascinating realm of specialized cells formed from stem cells, we uncover not only their remarkable functions, but also the stories of the scientists who have dedicated their lives to understanding them. The journey of stem cells, from their origins to their specialization, is a testament to the beauty of life itself. Each differentiated cell type, whether a neuron, cardiomyocyte, or blood cell, embodies the potential that lies within stem cells, waiting to be realized. Consider the implications of this potential. What if we could harness the power of stem cells to repair damaged tissues or regenerate lost function? Imagine a future where heart disease could be treated by regenerating damaged cardiomyocytes, or neurodegenerative diseases could be addressed by replenishing lost neurons. The possibilities are as vast as the ocean, driven by the diversity and adaptability of specialized cells. In the quiet moments when we reflect on the wonders of biology, it becomes clear that the story of life is, at its core, a story of transformation. From the humble beginnings of a single stem cell to the emergence of complex specialized cells, each step in this journey is infused with purpose and potential. The beauty of this transformation lies not only in the cells themselves, but in the profound impact they have on our lives, shaping our experiences and defining our existence. As we continue to explore this enchanting world, let us remain mindful of the interconnectedness of these specialized cells. Each one serves not only its own purpose, but also contributes to the broader narrative of health and vitality. The intricate dance of these cells is a reminder of the delicate balance that sustains life, urging us to appreciate the marvels of our own biology. In this journey through the realm of specialized cells, we can begin to understand the essential roles they play in our health and well-being. The neurons that help us think and feel, the cardiomyocytes that sustain our hearts, and the blood cells that protect us all exemplify the diversity that stem cells can produce. Each specialized cell type is a vital piece of the puzzle, contributing to the bigger picture of life itself. As we delve into their stories, we uncover not only the science, but also the beauty of existence, a tapestry woven from countless specialized cells, each one unique and irreplaceable. Within this vast biological landscape, the journey of stem cells does not end with the formation of neurons, cardiomyocytes, or blood cells. These specialized cells are merely the beginning of a remarkable story of adaptation and evolution, where each cell type is finely tuned to execute distinct functions that ensure the survival of the organism. Take, for instance, the epithelial cells that line our organs and tissues. These cells form protective barriers, shielding internal structures from pathogens and environmental factors. Imagine a fortress where each epithelial cell stands guard, meticulously orchestrating the exchange of substances while preventing unwanted intruders. The versatility of epithelial cells is astounding. They can specialize further into different forms, such as those found in the skin, lungs, or gastrointestinal tract. Each variation plays a crucial role in maintaining homeostasis, allowing the body to thrive in a myriad of conditions. Among the specialized cells derived from stem cells, muscle cells also deserve our attention. Beyond the cardiomyocytes of the heart, our bodies contain skeletal and smooth muscle cells, each possessing unique characteristics and functions. Skeletal muscle cells, for instance, are responsible for voluntary movements, enabling us to engage with the world around us. Picture the strength of a well-trained athlete, where each contraction of skeletal muscle is a testament to the intricate coordination of these specialized cells. Smooth muscle cells, found in organs such as the intestines and blood vessels, operate involuntarily, rhythmically contracting to facilitate digestion and regulate blood flow. The beauty of muscle cells lies in their ability to adapt to various demands, responding to signals from the nervous system and hormonal changes. Furthermore, consider the role of adipocytes, or fat cells, which are often overlooked in discussions of specialized cells. These cells serve not only as energy reservoirs, but also play essential roles in metabolic regulation and insulation. Imagine them as diligent caretakers, storing energy for times of need while also releasing hormones that influence appetite and metabolism. The balance of adipocytes is vital for overall health. An excess or deficiency can lead to metabolic disorders, reflecting the interconnectedness of specialized cells in maintaining equilibrium within the body. The diversity of specialized cells extends to the immune system as well, where various cell types work in concert to protect the body from infections. Beyond white blood cells, we find specialized immune cells, such as macrophages and dendritic cells, that play pivotal roles in recognizing and responding to pathogens. Macrophages, often regarded as the body's janitors, engulf and digest cellular debris and foreign invaders. Their vigilant presence is essential for maintaining tissue health and initiating inflammatory responses when necessary. Dendritic cells, on the other hand, act as messengers, capturing antigens and presenting them to other immune cells, thus orchestrating a coordinated defense. Each of these specialized cell types, whether they are epithelial cells, muscle cells, adipocytes, or immune cells, contributes to the symphony of life. They exemplify the profound importance of diversity within our cellular makeup. This diversity allows organisms to adapt to ever-changing environments, responding to challenges with resilience and grace. What if we pause to consider how this cellular diversity is not just a feature of biological systems, but a reflection of the interconnectedness of life itself? Each specialized cell is a unique note in the grand melody of health, where harmony and balance are essential for the organism's well-being. The intricate relationships among these cells highlight the importance of collaboration in maintaining homeostasis. Reflecting on the stories of individuals who have shaped our understanding of these specialized cells can provide inspiration. Think of the groundbreaking work of scientists like Elizabeth Blackburn, who explored the mechanisms of telomeres and their role in cellular aging, shedding light on how specialized cells can maintain their integrity over time. Her research has profound implications for our understanding of health, longevity, and the potential for cellular rejuvenation. As we explore the potential of stem cells further, we must also consider the implications of advancing our knowledge in regenerative medicine. Imagine a world where specialized cells can be replicated or repaired, allowing for the restoration of function in damaged tissues. The promise of stem cell therapies holds the potential to address conditions such as heart disease, neurodegenerative disorders, and injuries that result in loss of function. The future of medicine may very well hinge on our ability to harness the power of stem cells and their capacity to differentiate into the myriad of specialized cells that compose our bodies. In this unfolding narrative of specialized cells, we are reminded that each cell type is woven into the fabric of life, contributing to our health and vitality. The intricate connections between them form a complex network, a living testament to the beauty of biological diversity. As we journey deeper into the realm of specialized cells, let us remain mindful of the remarkable potential that lies within stem cells, waiting to be discovered and utilized for the betterment of human health. Imagine, if you will, the possibilities that lie ahead as we unlock the secrets of stem cell differentiation and specialization. The future is bright, filled with the promise of healing and regeneration, fueled by the incredible diversity and adaptability of the specialized cells that stem cells can create. Each discovery is a step closer to understanding the profound complexity of life, a reminder of the delicate balance that sustains us. Chapter 5: The Role of Stem Cells in Tissue Regeneration. The journey of healing and regeneration within our bodies is a testament to the resilience of life. Imagine, if you will, the intricate processes that unfold whenever tissue is damaged, whether from injury, disease, or the natural wear of time. Our bodies have a remarkable ability to repair themselves, and at the heart of this process lie stem cells, those extraordinary entities capable of transforming and adapting to meet the needs of our tissues. When an injury occurs, the body initiates a symphony of responses aimed at repairing the damage. The mechanisms of tissue repair and regeneration are multifaceted, involving not only the stem cells themselves, but also a network of signaling pathways and cellular interactions. Picture a bustling city, where construction crews mobilize swiftly to repair a damaged road. In a similar fashion, stem cells migrate to the site of injury, responding to signals released from damaged tissues. These signals act as beacons, guiding stem cells to their destination, where they can begin their transformative work. Stem cells contribute to the healing process primarily through their ability to differentiate into specialized cell types that are essential for tissue repair. This differentiation occurs in response to specific cues from their environment, allowing them to become the very cells that are needed to restore function. Imagine the versatility of these stem cells, akin to skilled artisans, each capable of crafting a unique structure, whether it be muscle, skin, or even nerve tissue. This remarkable adaptability is what makes stem cells so vital in the realm of regenerative medicine. Different tissues in the body employ distinct strategies for regeneration, often relying on their resident stem cells to facilitate repair. Take, for instance, the skin, the body's largest organ. Skin regeneration is a continuous process, with stem cells located in the basal layer of the epidermis constantly replenishing the outer layers of skin cells. These epidermal stem cells are pivotal in maintaining the integrity of the skin barrier, making it resilient against environmental damage. When an injury occurs, these stem cells spring into action, proliferating and differentiating to replace lost or damaged cells, ensuring that the skin can heal efficiently. Similarly, the blood is replenished through the magic of hematopoietic stem cells, which reside in the bone marrow. These cells are responsible for generating the various components of blood, including red blood cells, white blood cells, and platelets. Imagine the orchestrated effort required to maintain the delicate balance of these blood components. When an individual loses blood due to injury, hematopoietic stem cells quickly respond, producing new blood cells to restore circulation and support the immune system. Tissues such as the heart and nervous system present more complex challenges for regeneration. The heart, for instance, has a limited capacity for self-repair after an injury, such as a heart attack. Yet, recent research has shown that cardiac stem cells, although fewer in number than those in other tissues, play a crucial role in the regeneration of heart tissue. These cells can differentiate into cardiomyocytes, the heart muscle cells that are essential for proper function. The work of researchers like Eduardo Marbon has illuminated the potential for leveraging these cardiac stem cells to enhance heart repair and improve outcomes for patients. The nervous system, too, has its own intricacies when it comes to regeneration. Neurons, once damaged, have historically been viewed as incapable of regeneration. However, recent discoveries have revealed that certain types of neural stem cells exist within the brain and spinal cord. These cells possess the ability to regenerate neurons and glial cells, supporting the healing process in the central nervous system. The insights of pioneers like Fred Gage have shifted our understanding of neural regeneration, opening doors to new therapeutic possibilities for conditions such as spinal cord injuries and neurodegenerative diseases. Despite the promising potential of stem cells in tissue regeneration, there are notable limitations and challenges that researchers must navigate. One significant hurdle is the difficulty in harnessing the full potential of stem cells for therapeutic applications. While we are beginning to understand the mechanisms that govern stem cell behavior, much remains to be explored regarding how to effectively manipulate these cells in a clinical setting. Additionally, ethical considerations surrounding the use of embryonic stem cells continue to pose challenges within the field. The debate surrounding their use often centers on moral questions regarding the source of these cells and their implications. These discussions are necessary as we seek to balance scientific advancement with ethical responsibility. Moreover, the environment in which stem cells operate plays a crucial role in their effectiveness. The niche, or microenvironment, surrounding stem cells influences their behavior, differentiation, and overall function. Researchers are beginning to appreciate the importance of recreating these niches in laboratory settings to ensure that stem cells can thrive and perform their intended roles. This understanding emphasizes the need for innovative approaches in regenerative medicine, where the goal is not only to supply stem cells, but also to provide the right conditions for their success. As we look to the future of regenerative medicine, the potential for harnessing stem cells to treat a wide array of conditions is both exciting and daunting. The journey from understanding these cells to applying them effectively in clinical practice is still in its infancy. Yet, the stories of individuals pushing the boundaries of scientific knowledge continue to inspire hope. How might the next breakthroughs in stem cell research reshape our understanding of healing and regeneration? What new possibilities await us as we uncover the mysteries held within these remarkable cells? The quest for understanding stem cells and their role in tissue regeneration is a journey filled with promise and complexity. As we continue to delve into this fascinating field, we find ourselves standing at the precipice of a new era in medicine, where the extraordinary potential of stem cells may one day lead to transformative therapies that could redefine healing itself. As we explore the landscape of tissue regeneration, it is essential to understand the complexities of the healing environment that stem cells encounter. The microenvironment, often referred to as the niche, plays a pivotal role in guiding stem cell behavior. Imagine a garden where the soil quality, sunlight, and water availability dictate how plants grow and flourish. Similarly, the niche surrounding stem cells influences their ability to survive, proliferate, and ultimately differentiate into the necessary cell types for repair. Research has shown that various factors within the niche, including extracellular matrix components, oxygen levels, and the presence of signaling molecules, can dramatically affect stem cell function. This environment is not static. It changes in response to injury, creating a dynamic landscape that can either promote healing or hinder it. For instance, after an injury, the extracellular matrix may become enriched with specific proteins that signal for stem cell migration and activation. This orchestrated response is crucial as it enhances the regenerative capacity of tissues. Consider the role of growth factors, which act as chemical messengers that modulate stem cell behavior. Certain growth factors, such as fibroblast growth factors and transforming growth factor beta, are released during the healing process to stimulate stem cells. Picture these factors as guiding lights, illuminating the path for stem cells to follow as they navigate the complexities of tissue repair. By binding to receptors on the surface of stem cells, these growth factors can initiate a cascade of signaling events, prompting cells to divide or differentiate based on the specific needs of the tissue. However, the journey of stem cells in the realm of regeneration is not without its challenges. One significant limitation lies in the aging process. As organisms age, the regenerative potential of stem cells tends to diminish. This decline can be likened to a once vibrant garden that, over time, fails to produce as many blooms. The reasons behind this decline are multifaceted, involving changes in the niche, increased inflammation, and alterations in the intrinsic properties of the stem cells themselves. For instance, aged stem cells may exhibit reduced proliferation rates or impaired differentiation capacity, making it more difficult for tissues to heal effectively. The heart serves as a poignant example of these challenges. While cardiac stem cells are present, their limited abundance and functionality decrease with age. This reduction complicates the heart's ability to recover from injuries such as myocardial infarctions. The work of researchers, including those studying the effects of exercise and lifestyle on stem cell health, has highlighted the potential for interventions that may rejuvenate stem cells and restore their regenerative capabilities. How might the choices we make in our daily lives influence the vitality of these cells? Another challenge in the field of regenerative medicine is the risk of uncontrolled growth. Stem cells possess the remarkable ability to divide indefinitely, leading to concerns about tumorigenesis. When stem cells are manipulated or introduced into a damaged tissue, the delicate balance of growth and differentiation must be carefully managed. Imagine sculpting a statue from a block of marble. Too much force could lead to irreversible damage. Researchers are acutely aware of the need to ensure that stem cell therapies promote healing without the unintended consequence of tumor formation. Moreover, the field faces hurdles in translating laboratory discoveries into clinical applications. While the possibilities of stem cell therapies are vast, the path to effective treatment is often fraught with regulatory, ethical, and technical challenges. The translation from bench to bedside requires rigorous testing and validation to ensure safety and efficacy. As researchers strive to bring new therapies to fruition, they must navigate a landscape filled with uncertainties, yet the promise of regenerative medicine continues to inspire them. The potential for stem cells to revolutionize medicine carries with it a sense of urgency. What if we could harness the power of stem cells not only to heal injuries, but also to combat degenerative diseases? The visions of scientists like Shinya Yamanaka, who discovered induced pluripotent stem cells, offer a glimpse into this future. By reprogramming adult cells to a pluripotent state, researchers have opened a new frontier in regenerative medicine, allowing for a broader array of therapeutic possibilities. This breakthrough has sparked a wave of enthusiasm and innovation, igniting conversations about personalized medicine and the ability to create patient-specific cell types for treatment. As we ponder these advancements, we are left to consider the ethical implications that arise. How do we strike a balance between innovation and responsibility? The discourse surrounding stem cell research is complex, encompassing concerns about consent, the source of stem cells, and the potential for exploitation. Engaging in these discussions is essential as we forge ahead into an era where the possibilities seem boundless. The journey through the realm of stem cells and tissue regeneration is a tale of resilience, complexity, and hope. As we continue to unravel the mysteries of these remarkable cells, we find ourselves standing on the cusp of a medical revolution. The stories of those who dare to dream and explore the unknown remind us that within the challenges lie the seeds of discovery. What new horizons might stem cell research unveil in the quest for healing? With each step forward, we draw closer to a future in which the power of regeneration may hold the key to overcoming some of humanity's most pressing health challenges. Chapter 6: Applications of Stem Cell Research in Medicine. In the realm of modern medicine, stem cell research emerges as a beacon of hope, illuminating paths toward healing and recovery. Today, a variety of stem cell therapies are already in use, showcasing their transformative potential across diverse medical fields. The application of stem cells has taken many forms, from treating blood disorders like leukemia through hematopoietic stem cell transplants to pioneering therapies for conditions such as spinal cord injuries. Imagine a world where damaged tissues can be repaired or replaced, where hope is not merely a fleeting sentiment, but a tangible reality rooted in scientific innovation. One of the most promising areas of stem cell therapy is the treatment of degenerative diseases. Conditions such as Parkinson's disease, Alzheimer's disease, and diabetes represent significant challenges in modern healthcare. These diseases often arise from the gradual loss of specific cell types, leading to the disruption of normal bodily functions. Picture the delicate balance of a finely tuned instrument, where the absence or malfunction of even a single component can disrupt the entire symphony. Stem cells, with their remarkable ability to differentiate into various specialized cells, offer a solution to restore this balance. For instance, researchers are exploring the potential of using neural stem cells to regenerate lost neurons in the brains of Parkinson's patients. The prospect of restoring lost function and improving quality of life is a powerful motivator for scientists and medical professionals alike. Yet, with great promise comes the weight of ethical considerations. The discourse surrounding stem cell research is often fraught with complex moral dilemmas. The use of embryonic stem cells, in particular, has sparked significant debate. Some argue that the potential benefits of regenerative medicine justify the use of these cells, while others hold firm beliefs about the sanctity of human life from its earliest stages. This conflict raises important questions. How do we balance scientific advancement with ethical responsibility? What frameworks can we establish to ensure that stem cell research is conducted with integrity and respect for human dignity? Many researchers advocate for the use of adult stem cells or induced pluripotent stem cells as alternatives, which may circumvent some of the ethical concerns associated with embryonic sources. Induced pluripotent stem cells, for example, are reprogrammed from adult cells, allowing for the creation of pluripotent cells without the ethical implications tied to human embryos. This development is akin to finding a new route through a dense forest, illuminating the way forward without compromising our moral compass. Looking to the horizon, the future of stem cell applications in healthcare gleams with promise. As our understanding of stem cells deepens, new possibilities emerge. Researchers are investigating the integration of stem cells into personalized medicine, tailoring treatments based on individual genetic profiles. Imagine a future where therapies are designed specifically for you, optimizing healing and minimizing side effects. This shift represents a significant transformation in how we approach healthcare, moving from a one-size-fits-all model to one that recognizes the uniqueness of every individual. Moreover, advancements in biotechnology are paving the way for more effective and less invasive delivery methods for stem cell therapies. Techniques such as bioengineering scaffolds and 3D bioprinting of tissues are at the forefront of this innovation. Visualize a future where damaged organs can be replaced with lab-grown equivalents, crafted precisely to match the recipient's anatomy. This dream is no longer confined to the pages of science fiction. It is progressively becoming a reality. In the tapestry of medicine, the integration of stem cell research is not merely about the cells themselves, but the stories of the individuals they touch. Each breakthrough holds the potential to change lives, to restore hope where it was once lost. As we stand on the brink of a new era in medicine, the call to harness the power of stem cells echoes louder than ever. What stories will unfold as we continue this journey? What lives will be transformed by our relentless pursuit of knowledge and healing? The answers remain to be seen, yet the path is illuminated by the promise of discovery and the unwavering spirit of human resilience. The journey of stem cell therapy is not just limited to the treatment of diseases. It also extends into the realm of regenerative medicine, where the potential to heal injuries and restore function is being explored with fervor. Imagine a future where a simple injection of a patient's own stem cells can mend a torn ligament or rejuvenate damaged heart tissue. This vision is becoming increasingly plausible as research unveils the remarkable properties of stem cells in promoting healing and regeneration. One of the most striking examples of this potential is seen in the field of cardiac repair. Heart disease remains a leading cause of mortality worldwide, often resulting in irreversible damage to heart tissues. Scientists are investigating the use of stem cells to repair this damage and restore heart function. By harnessing the regenerative capabilities of stem cells, researchers hope to regenerate healthy cardiac tissue, offering new hope to those suffering from heart failure. Imagine the impact of a therapy that could not only extend life, but also significantly enhance the quality of life for millions. Furthermore, the application of stem cell therapy in orthopedics has gained traction, particularly in the treatment of joint injuries and cartilage degeneration. Stem cells can be harvested from various tissues, including bone marrow and adipose tissue, and injected directly into damaged joints. These cells can then differentiate into cartilage-producing cells, effectively repairing the wear and tear that often accompanies aging or injury. This approach not only alleviates pain, but also restores mobility, allowing individuals to engage in activities they once thought lost forever. The concept of using stem cells for tissue engineering is also captivating. Picture a laboratory filled with scientists meticulously crafting bioengineered tissues, replicating the intricate architecture of human organs. In this scenario, stem cells serve as the foundational building blocks, guiding the formation of functional tissues that can be implanted into patients. This innovative approach has the potential to address the critical shortage of donor organs, transforming the landscape of transplantation. The idea that one day we could grow organs in a lab tailored to fit individual patients is no longer a distant dream. It is a burgeoning reality. As we explore the vast potential of stem cell applications, one cannot overlook the ethical considerations that accompany such advancements. While the promise of regenerative medicine is alluring, it raises profound questions about the implications of manipulating life at such fundamental levels. The debate surrounding the ethics of stem cell research continues to evolve, with discussions around the sources of stem cells, informed consent, and the long-term consequences of these therapies dominating the conversation. One prominent figure in the field, who has often emphasized the need for ethical reflection in scientific endeavors, stated, "Science and ethics are not mutually exclusive, they are two sides of the same coin." This sentiment resonates deeply as we navigate the complexities of stem cell research. Ethical guidelines are essential in ensuring that the advancements made in science do not outpace our moral responsibilities. The establishment of ethical frameworks can guide researchers, policymakers, and society in making informed decisions about the direction of stem cell research and its applications. The landscape of stem cell therapy is also shaped by the cultural and societal perceptions of science. There are communities that embrace the innovations in regenerative medicine with open arms, while others harbor fears and skepticism. It is crucial to foster open dialogues that bridge the gap between scientific advancements and public perception. By engaging the community in discussions about the benefits and risks of stem cell therapies, we can cultivate a more informed public, ready to embrace the potential of this transformative field. As we look to the future, the possibilities for stem cell applications in healthcare seem limitless. Emerging technologies, such as gene editing and advanced biomaterials, are converging with stem cell research to create groundbreaking therapies that were once thought impossible. The intersection of these disciplines holds the promise of personalized medicine at its finest. Imagine therapies that not only repair damage, but also target the underlying genetic causes of diseases. The implications for conditions ranging from hereditary disorders to cancer are profound, introducing a new era of precision medicine. Moreover, the continued exploration of stem cells in the context of aging is another frontier that beckons. As the global population ages, the need for effective interventions to combat age-related degenerative diseases becomes increasingly urgent. Researchers are delving into how stem cells can be utilized to rejuvenate aging tissues, potentially extending not just lifespan, but health span, the period of life spent in good health. This approach could redefine our understanding of aging and empower individuals to lead vibrant, active lives well into their later years. The excitement surrounding stem cell research is palpable, and the dedication of scientists, clinicians, and advocates fuels the momentum. Each discovery brings us closer to unlocking the secrets of stem cells, whether through novel therapies for existing conditions or innovative applications that redefine our understanding of healing. How many lives could be changed through these efforts? The potential is staggering, and as we continue to explore this uncharted territory, the promise of stem cells remains a testament to human ingenuity and the relentless pursuit of health and wellness. In this ongoing journey, the stories of those affected by stem cell therapies serve as powerful reminders of the impact of scientific exploration. From patients who have regained mobility after debilitating injuries to those who have experienced remarkable improvements in their quality of life, each narrative echoes the profound significance of stem cell research. What new stories will emerge as we venture further into this landscape? With every advancement, we inch closer to realizing the incredible possibilities that stem cells hold for the future of medicine. As the journey of stem cell research advances, the integration of these remarkable cells into therapeutic practices unfolds even further. Beyond the immediate applications in regenerative medicine, there is a growing interest in how stem cells can influence the treatment of various types of cancers. Cancer arises from the uncontrolled growth of cells, often due to genetic mutations and environmental factors. Imagine the complexity of a garden where weeds proliferate amidst the flowers, threatening to overtake the beauty of the landscape. In this metaphor, stem cells hold the potential to be the gardeners, selectively targeting and eliminating the invasive cells that disrupt harmony. Researchers are exploring the role of stem cells in developing targeted therapies that could specifically attack cancer cells while preserving healthy tissues. One intriguing approach involves utilizing stem cells as delivery vehicles for cancer-fighting agents, effectively turning them into smart missiles that home in on tumors. This innovative strategy emphasizes the adaptability of stem cells, which can be engineered to express specific receptors that recognize and bind to cancerous cells. Consider the possibilities of this technology. It could revolutionize how we approach cancer treatment, minimizing the debilitating side effects often associated with conventional therapies. Moreover, the adaptability of stem cells is not limited to their application in targeted therapies. Investigations into the tumor microenvironment have revealed how stem cells can potentially influence the behavior of cancer cells. They can produce signaling molecules that alter the local environment, either promoting or inhibiting tumor growth. This dual role as both a therapeutic agent and a modulator of tumor dynamics invites a deeper understanding of the complex interplay between stem cells and cancer biology. As we contemplate the future, the prospects for stem cells within the realm of personalized medicine continue to expand. The concept of tailoring treatments to individual genetic profiles is becoming a reality. Imagine a world where each patient's specific genetic makeup informs the selection of therapies, enhancing efficacy and reducing adverse effects. This shift towards precision medicine not only embodies a more humane approach to healthcare, but also signifies a profound understanding of the relationship between genetics and disease. The journey into this new frontier raises compelling questions. How can we ensure that these advancements are accessible to all? As we develop personalized therapies, it is vital to consider the ethical implications of such tailored treatments. Will they widen the gap between those who can afford innovative therapies and those who cannot? Addressing these concerns is paramount as we strive to create an equitable healthcare system that honors the dignity of every individual. In the arena of stem cell research, the exploration of ethical considerations remains a guiding principle. The dialogue surrounding the use of stem cells must evolve alongside scientific advancements. Innovative solutions, such as the development of biobanks and registries, can provide transparency and foster trust within communities. By engaging stakeholders in discussions about the benefits and risks associated with stem cell therapies, researchers can cultivate a shared understanding that resonates with the public. Another exciting area of exploration lies in the intersection of stem cells and bioengineering. As scientists delve into the realm of synthetic biology, the possibility of creating artificial tissues and organs using stem cells becomes increasingly feasible. Imagine a scenario where damaged organs could be replaced with bioengineered alternatives, crafted from a patient's own cells to reduce the risk of rejection. This paradigm shift in organ transplantation could alleviate the burdens of waiting lists and enhance the quality of life for countless individuals. As we ponder the implications of these advancements, it is essential to remain grounded in the ethical considerations that accompany such powerful technologies. The ability to create lifelike tissues raises profound questions about the nature of life and the responsibilities that come with wielding such capabilities. Engaging in reflective practices and ethical discussions can help navigate these complexities, ensuring that the pursuit of scientific knowledge remains firmly anchored in human values. In the landscape of stem cell research, the stories emerging from ongoing clinical trials provide a glimpse into the transformative impact of these therapies. Patients who participate in these trials often embody hope, courage, and resilience, serving as living testaments to the potential of scientific exploration. Each success story reflects not only the advancements in medicine, but also the profound connections forged between patients and the healthcare community. As we stand at this crossroads, the future of stem cell applications in medicine beckons us to embrace the unknown with curiosity and compassion. How will our understanding of stem cells continue to evolve? What breakthroughs lie ahead, waiting to be uncovered? While the answers remain elusive, the journey promises to be rich with discovery, illuminating the path toward healing and renewal for generations to come. Chapter 7: Challenges in Stem Cell Differentiation Research. In the realm of stem cell research, where promise meets complexity, scientists face a multitude of challenges that can hinder progress towards effective therapies. One significant hurdle lies in the technical obstacles associated with manipulating stem cells. Imagine, for a moment, the delicate nature of these cells, akin to fine porcelain, requiring careful handling to maintain their integrity. Researchers must design and implement precise techniques to coax these stem cells into desired forms, a process that demands not only expertise, but also an understanding of the intricate biological environment they inhabit. The manipulation of stem cells often involves complex procedures, including genetic engineering and the use of growth factors that can influence their behavior. Each type of stem cell reacts differently to these manipulations, introducing variability into the process. Picture a painter selecting colors for a canvas. The choice of pigment can drastically alter the final image. Similarly, the specific techniques and conditions used to manipulate stem cells can lead to vastly different outcomes in terms of differentiation, growth rates, and stability. This variability in differentiation outcomes is a core challenge faced by researchers. Not all stem cells respond uniformly to the same stimuli, leading to an unpredictable array of specialized cells. Imagine planting seeds in a garden. While you may expect a certain flower to bloom, sometimes unexpected varieties surprise you. In stem cell research, this unpredictability can complicate the efforts to develop therapies, as scientists strive for consistency and reliability in outcomes. Moreover, the pathways that govern stem cell differentiation are influenced by a multitude of factors, including the cellular environment and the presence of specific signaling molecules. Each stem cell type has its own repertoire of responses, making it essential for researchers to explore and understand these mechanisms thoroughly. The path from stem cell to specialized cell is not a straight line. Rather, it resembles a winding road, filled with detours and unexpected turns. Addressing safety concerns with stem cell therapies presents another layer of complexity. The remarkable potential of stem cells to regenerate and repair tissues comes with inherent risks. When stem cells differentiate, there is a possibility that they may form unintended cell types, leading to complications such as tumor formation. Imagine a sculptor carving a statue from marble who must be cautious not to chip away too much, lest the creation becomes something unrecognizable. Similarly, researchers must tread carefully, ensuring that the pathways activated during differentiation do not lead to malignant transformations. The safety of stem cell therapies is paramount, and as scientists venture into the clinic, they must balance the potential benefits with the risks involved. This necessitates rigorous testing and evaluation. Picture a ship at sea, navigating through stormy waters. The captain must be vigilant, assessing the winds and currents to ensure a safe passage. In the same vein, researchers must meticulously evaluate the safety of their therapies before they are administered to patients, often involving preclinical studies and extensive trials. Compounding these challenges are the regulatory hurdles that researchers face. The landscape of stem cell research is shaped by a complex regulatory framework designed to protect patients and ensure the ethical application of these therapies. Imagine a labyrinth where each turn requires careful consideration and understanding of the rules that govern entry and exit. Researchers must navigate this maze, seeking approval for their work while adhering to stringent guidelines that can vary by region and country. Regulatory bodies require comprehensive documentation, demonstrating not only the efficacy of a proposed therapy, but also its safety and ethical considerations. This process can be time-consuming and resource-intensive, often delaying the transition from the laboratory to the clinic. Scientists may find themselves in a race against time, where the clock ticks away while they gather the necessary data to satisfy regulatory requirements. The path to approval can feel daunting, yet it is essential for maintaining public trust and ensuring that stem cell therapies are safe for widespread use. Amidst these challenges, the perseverance of researchers shines through. Their determination is reminiscent of pioneers in other fields who faced skepticism and setbacks, but pushed forward in the name of discovery. Scientists such as Shinya Yamanaka, who was awarded the Nobel Prize for his work in reprogramming adult cells into pluripotent stem cells, exemplify the spirit of innovation that drives progress in this field. Yamanaka once stated that the future of medicine will be determined by how well we understand and harness the power of stem cells. As researchers continue to tackle these multifaceted challenges, they draw upon the lessons learned from past successes and failures. Each experiment, each setback, offers insights that contribute to a deeper understanding of stem cell biology. Picture an artist refining their technique over time, each brush stroke a step toward mastery. In the same way, scientists refine their approaches, learning from the variability and complexities inherent in stem cell differentiation. In this ongoing pursuit, collaboration emerges as a key component. Researchers from diverse disciplines come together, pooling their expertise to address the many obstacles that lie in wait. The integration of knowledge from molecular biology, genetics, and bioengineering creates a rich environment for innovation. Imagine a symphony composed of various instruments, each contributing its unique sound to create a harmonious masterpiece. Collaboration in stem cell research allows for the sharing of ideas and resources, ultimately leading to more robust and effective solutions. As we reflect on the challenges in stem cell differentiation research, one cannot help but feel a sense of awe at the resilience of the human spirit. Each obstacle faced is met with determination and creativity, pushing the boundaries of what is known and what is possible. The journey of stem cell research is as much about discovery as it is about overcoming the hurdles that stand in the way. The potential of stem cells is vast, and the road ahead, while fraught with challenges, is illuminated by the promise of healing and regeneration. As researchers continue to refine their techniques, address safety concerns, and navigate regulatory landscapes, they forge ahead with unwavering commitment. Each step taken in this field brings us closer to realizing the full potential of stem cells, transforming not just the landscape of medicine, but also the lives of countless individuals in need of innovative therapies. As researchers delve into the complexities of stem cell differentiation, they encounter a labyrinth of technical obstacles that can impede their progress. The variability in outcomes during differentiation remains a persistent challenge. Imagine a symphony orchestra, where each instrument, while capable of producing beautiful music, requires a skilled conductor to harmonize the performance. In the same way, the differentiation of stem cells requires precise conditions and techniques to yield consistent results. The inherent variability in stem cell behavior can stem from numerous factors, including genetic background, the source of the stem cells, and the specific culture conditions employed. Think of a recipe where substituting one ingredient can lead to a completely different dish. Researchers must carefully balance these factors, ensuring that the conditions they create mimic the natural environment of the cells as closely as possible. This balance is critical, as even slight deviations can lead to unexpected results, complicating the path toward effective therapies. The intricacies of manipulating stem cells extend beyond mere technical challenges. As scientists work to guide these cells toward specific fates, they must navigate a delicate dance of signaling pathways and cellular interactions. These processes are often subject to influence from the surrounding microenvironment, which can modify how stem cells respond to cues. Imagine a flower growing in a diverse ecosystem. Its growth and development will heavily depend on the soil, sunlight, and rainfall available. Similarly, stem cells are influenced by their surroundings, underscoring the need for researchers to create optimal conditions for differentiation. As the journey of stem cell differentiation unfolds, safety concerns emerge as a prominent issue. The potential for stem cells to form unintended cell types raises red flags regarding their use in clinical applications. Picture a gardener tending to a delicate plant, ensuring that it grows into the intended form without the risk of becoming invasive. Researchers must embrace a cautious approach, rigorously studying the differentiation pathways to prevent malignancies and other complications. This vigilance is essential, as the stakes are high when it comes to patient safety. The road to understanding the full potential of stem cell therapies is paved with regulatory hurdles. Each country and region has its own rules governing stem cell research, often shaped by ethical considerations and safety protocols. Imagine an artist seeking to showcase their work in a gallery, only to find that each venue has different standards for exhibition. Researchers must adapt to these varying regulations, often investing significant time and resources to ensure compliance while pursuing their vision of therapeutic innovation. To navigate these challenges, interdisciplinary collaboration is vital. Scientists from various fields, including molecular biology, bioengineering, and ethics, converge to address the complexities inherent in stem cell research. This collaborative spirit fosters a rich exchange of ideas, allowing researchers to approach problems from diverse angles. Picture a group of explorers, each with their unique skills, joining forces to tackle an uncharted territory. Together, they enhance the potential for breakthroughs by pooling their knowledge and expertise. As scientists continue to confront these multifaceted challenges, they remain motivated by the profound possibilities that stem cell research presents. The stories of pioneering figures, such as James Thomson, who first isolated human embryonic stem cells, inspire a generation of researchers to push the boundaries of science. Thomson's work opened doors to unexplored avenues and demonstrated the immense potential of stem cells in advancing medicine. His journey, marked by determination and curiosity, reminds us that every obstacle can be transformed into an opportunity. The quest for effective stem cell therapies is not merely a scientific endeavor. It is a deeply human one. The potential to alleviate suffering and restore health resonates with researchers and patients alike. Each new discovery, no matter how small, brings hope to those who await innovative treatments. Imagine a light illuminating a dark path, guiding individuals toward healing and recovery. This hope fuels the relentless pursuit of knowledge, urging scientists to overcome the challenges before them. In the face of adversity, researchers remain steadfast, drawing inspiration from the resilience of the very stem cells they study. These remarkable cells embody the essence of adaptability and potential, showcasing the power of transformation. Just as stem cells can develop into various specialized cells, so too can researchers adapt their strategies in the face of challenges. This parallel serves as a reminder that progress in science is not always linear. It requires perseverance, creativity, and a willingness to learn from each experience. As the landscape of stem cell research continues to evolve, so too do the challenges and opportunities it presents. The commitment to understanding the intricacies of stem cell differentiation, while addressing safety and regulatory concerns, is a collective mission that unites researchers worldwide. This journey of discovery is marked by both triumphs and setbacks, yet it is propelled forward by an unwavering belief in the transformative power of stem cells. Imagine a future where the challenges of today have been overcome, where stem cell therapies are not just a dream but a reality, accessible to all who need them. This vision drives scientists to persist, to innovate, and to collaborate, forging new pathways toward healing. Each step taken in this pursuit brings us closer to unlocking the full potential of stem cells, offering hope and healing to those who seek it most. The challenges may be formidable, but so too is the spirit of discovery that fuels this remarkable journey. Chapter 8: Innovations in Stem Cell Technology. In the ever-evolving landscape of stem cell research, a wave of innovation has emerged, transforming the way scientists culture stem cells, unlocking new potentials for medical advancements. Historically, stem cell culture techniques relied heavily on the use of specific growth factors and substrates, often leading to variability in the behavior and characteristics of the cultured cells. However, recent advances have introduced new methodologies that promise greater consistency and efficiency. Imagine a serene garden, where each plant thrives in a carefully designed environment, nourished by the right balance of light, water, and nutrients. In a similar manner, contemporary culture techniques utilize sophisticated scaffolds and bioreactor systems that mimic the natural extracellular matrix, providing the ideal conditions for stem cells to flourish and maintain their pluripotency. These advancements are complemented by the integration of three-dimensional culture systems, offering a more accurate representation of in vivo environments. Traditional two-dimensional cultures often fail to capture the complexities of cellular interactions and tissue architecture, but three-dimensional cultures allow cells to organize themselves more naturally. Picture a bustling city where buildings rise in intricate formations, each contributing to the collective structure. In three-dimensional cultures, stem cells can form structures akin to tissues and organs, enhancing our understanding of their behavior and functions. This innovative approach is not merely a technical upgrade. It opens doors to profound discoveries. Researchers can now study the intricacies of stem cell differentiation and the microenvironments that influence this process. The ability to observe these cells in more lifelike conditions provides insights into how they respond to various stimuli, including drugs or genetic modifications. Imagine standing at the forefront of a new era in science, where the secrets of cellular behavior are unveiled in real time, leading to breakthroughs that were once thought unattainable. At the intersection of stem cell research and genetic engineering lies CRISPR technology, a revolutionary tool that has garnered immense attention and transformed the field in recent years. CRISPR, short for clustered regularly interspaced short palindromic repeats, empowers scientists to edit genes with remarkable precision. This technique can be likened to a word processor, allowing researchers to edit the genetic code of an organism, correcting errors or introducing new traits. The implications for stem cell research are staggering. Imagine being able to target and modify specific genes within stem cells, enhancing their potential for therapeutic applications. One of the most significant applications of CRISPR in stem cell research is the correction of genetic disorders. By repairing faulty genes in stem cells derived from patients, scientists can potentially develop personalized therapies that address the root cause of diseases. Consider the story of a young patient suffering from a genetic condition that affects their ability to produce certain enzymes. With CRISPR, researchers can modify the patient's own stem cells, allowing them to grow healthy cells that produce the necessary enzymes. This approach not only offers hope for treatment, but also represents a shift towards personalized medicine, tailoring therapies to the unique genetic makeup of individuals. Moreover, CRISPR can be employed to create disease models within stem cells, providing invaluable insights into the mechanisms of various disorders. Imagine a laboratory bustling with activity, where scientists utilize modified stem cells to reproduce the conditions of diseases like Alzheimer's or diabetes. By observing these models, researchers can better understand disease progression, identify new therapeutic targets, and test potential treatments in a controlled environment. As the field of stem cell technology continues to expand, the development of organoids has emerged as a groundbreaking innovation. Organoids are miniature, simplified versions of organs that are grown in vitro from stem cells. These tiny structures recapitulate key features of actual organs, providing a unique platform for research and drug testing. Picture a small, thriving ecosystem, where each organoid serves as a microcosm of its larger counterpart, offering insights into organ function, development, and disease. The significance of organoids lies in their ability to bridge the gap between basic research and clinical applications. By studying organoids derived from human stem cells, researchers can gain a deeper understanding of human biology and pathology. This is particularly important in the context of diseases that are difficult to model in traditional animal studies, as organoids provide a more relevant and ethical alternative. Imagine witnessing the inner workings of a human liver or brain, observing cellular interactions and responses to treatments in real time, all without the ethical concerns that come with animal models. Organoids also present a powerful tool for drug development and personalized medicine. By creating organoids from patients' own cells, scientists can test the efficacy and toxicity of various compounds, tailoring treatments to individual responses. This approach holds the potential to revolutionize the way we develop and prescribe medications, ensuring that patients receive therapies that are not only effective but also safe. The collaboration between research institutions and industry has played a pivotal role in driving these innovations within stem cell technology. As scientists push the boundaries of what is possible, partnerships with biotechnology and pharmaceutical companies facilitate the translation of discoveries into practical applications. Imagine a bridge connecting the realms of academia and industry, where ideas flow freely and innovation thrives. These collaborations harness the strengths of both sectors, combining scientific expertise with the resources and infrastructure needed to bring new therapies to market. By working together, researchers and industry leaders can streamline the process of developing stem cell-based therapies, ensuring that breakthroughs reach patients more swiftly. One compelling example of such collaboration is the partnership between academic institutions and biotech firms focused on regenerative medicine. By leveraging cutting-edge research and advanced manufacturing techniques, these partnerships aim to produce scalable and affordable stem cell therapies. Picture a vibrant marketplace of ideas and solutions, where the latest advancements in stem cell technology are not only explored in laboratories, but also brought to the forefront of clinical practice. Furthermore, collaborations can facilitate the sharing of knowledge and resources, allowing researchers to access advanced technologies and methodologies that may not be available within their institutions. This collaborative spirit fosters an environment of innovation, where ideas can flourish and groundbreaking discoveries can emerge. As we explore the realm of innovations in stem cell technology, it becomes clear that the future holds immense promise. The advances in stem cell culture techniques, the power of CRISPR and gene editing, the emergence of organoids, and the fruitful collaborations between research and industry all contribute to a landscape rich with potential. Each of these elements represents a thread in the intricate fabric of scientific advancement, weaving together a narrative of hope and healing. The journey of discovery continues, propelled by curiosity, creativity, and a shared commitment to unlocking the mysteries of stem cells for the betterment of humanity. The advances in stem cell culture techniques have paved the way for innovative approaches in research, yet they are only part of a larger narrative in the realm of cellular biology. As we consider the intricate possibilities that lie ahead, we must delve into the exciting world of organoids. These miniature organ systems have emerged as a remarkable innovation, revolutionizing not just our understanding of human biology, but also the avenues through which we can approach drug testing and disease modeling. Organoids, formed from stem cells, replicate the architecture and functionality of actual organs. Imagine a tiny, pulsating heart or a miniature brain, each crafted from the very cells that hold the potential to regenerate and heal. These three-dimensional structures are not merely facsimiles. They exhibit the complex cellular interactions and tissue organization found in their larger counterparts. The ability to culture organoids provides researchers with a platform to study organ development and disease processes in ways that traditional two-dimensional cell cultures cannot. Take, for instance, the liver organoid. When derived from stem cells, these organoids can mimic various liver functions, offering insights into metabolic processes, liver disease, and drug metabolism. This is particularly significant given the liver's role in detoxifying harmful substances and metabolizing medications. Envision a laboratory where scientists observe these organoids reacting to different drugs, providing critical data that could lead to safer and more effective therapies. The significance of organoids extends beyond their immediate utility in research. They serve as a bridge connecting the realms of basic science and clinical application. Imagine a patient suffering from a rare genetic disorder. Researchers can create organoids from the patient's own stem cells, allowing them to study the disease mechanism in a controlled environment. This not only enhances understanding, but fosters the development of personalized treatment strategies tailored to the individual's unique genetic makeup. In the backdrop of these advancements, the role of CRISPR and gene editing techniques cannot be overlooked. The fusion of CRISPR technology with stem cell research has opened new frontiers in our pursuit of understanding and treating genetic disorders. This powerful tool allows scientists to precisely edit genes, correcting mutations that give rise to various diseases. Consider the implications of this for conditions like cystic fibrosis or sickle cell disease, where a single genetic mutation can have profound effects on the individual's health. With CRISPR, researchers can target these mutations in stem cells, effectively restoring normal function. Imagine the hope for families affected by genetic disorders as the prospect of personalized therapies becomes increasingly tangible. The ability to edit genes with pinpoint accuracy not only enhances the therapeutic potential of stem cells, but also raises ethical questions about the extent of our intervention in the natural order of life. At the same time, the collaboration between research institutions and industry has become a crucial element in the advancement of stem cell technologies. As scientists uncover groundbreaking discoveries, partnerships with biotech companies facilitate the translation of these findings into clinical applications. Picture a thriving ecosystem where academia and industry converge, each lending its strengths to accelerate the path from laboratory research to patient care. These collaborations are vital for several reasons. They enable researchers to access cutting-edge technologies and resources that enhance their work. For instance, the incorporation of advanced imaging techniques allows scientists to visualize the behavior of stem cells and organoids in real time. Imagine the excitement of observing cellular processes unfold before your eyes, revealing insights that were previously obscured. Moreover, these partnerships streamline the development of stem cell-based therapies, ensuring that innovations reach those in need more swiftly. In today's fast-paced scientific landscape, the ability to move from discovery to application is paramount. Collaborations can help bridge the gap between theoretical research and practical implementation, allowing for a more comprehensive approach to healthcare challenges. The confluence of advances in stem cell culture techniques, CRISPR technology, the development of organoids, and collaborative efforts between research and industry paints a vivid picture of the future. Each innovation feeds into the next, creating a vibrant network of possibilities that hold the promise of transformative therapies. As we envision this future, we find ourselves standing at the precipice of a new era in medicine, where the mysteries of stem cells are gradually being unraveled, and where the potential for healing extends far beyond what was once thought possible. Imagine the day when conditions previously deemed untreatable become manageable, or when personalized therapies are the norm rather than the exception. This vision is not merely a dream, but a tangible reality on the horizon, driven by the relentless pursuit of knowledge and the unwavering commitment to improve human health. The journey of discovery is ongoing, and as we continue to explore the depths of stem cell technology, we are reminded of the profound impact that these innovations will have on our lives and the lives of future generations. Chapter 9: The Future of Stem Cell Therapy. As we gaze into the horizon of stem cell therapy, a landscape rich with possibilities unfurls before us. Imagine a world where diseases that once seemed insurmountable can be treated with the very cells that form the foundation of our being. The future of stem cell applications is not only promising, but also poised to redefine the boundaries of medicine. Predictions abound of remarkable advancements that will enhance our ability to harness the power of stem cells, leading to therapies that can profoundly impact chronic conditions that plague humanity. Consider the potential of stem cells in managing chronic diseases such as heart disease, diabetes, and neurodegenerative disorders. For instance, researchers are exploring innovative techniques to regenerate damaged heart tissue following a heart attack. Picture this: a patient's own stem cells are harvested, coaxed into becoming cardiac muscle cells, and then reintroduced into the heart. The goal is to restore function, heal damage, and enhance quality of life. This paradigm shift in treatment could mean that one day, heart failure may be managed not with medications alone, but with regenerating the very tissue that has suffered. In addition to heart disease, the horizon gleams with hope for conditions like type 1 diabetes. Imagine a future where patients no longer need to rely on insulin injections. Instead, stem cells could be used to generate insulin-producing beta cells, restoring normal blood sugar regulation. The idea of a therapy that could eliminate the need for constant monitoring and medication is tantalizing and speaks to the transformative power of stem cells. The integration of personalized medicine with stem cell treatments is another aspect that promises to revolutionize healthcare. Think of a treatment plan tailored to the individual, one that considers their unique genetic makeup and health history. Stem cells offer a pathway to create personalized therapies that not only address the condition, but also resonate with the specific biological nuances of each patient. This approach could lead to more effective treatments with fewer side effects, as therapies can be designed to work harmoniously with the individual's body. As we venture into this realm, the notion of precision medicine becomes paramount. The mapping of the human genome has opened doors to understanding diseases at a molecular level. Imagine the possibilities of combining this genetic information with stem cell therapies, creating a new frontier in disease management. Researchers envision not only treating conditions after they arise, but also preemptively addressing them by understanding the genetic predispositions individuals carry. However, with great potential comes great responsibility. The ethical frameworks governing stem cell research and application must evolve alongside scientific advancements. As we step into this future, questions arise regarding the moral implications of using stem cells, particularly those derived from embryonic sources. It is imperative to consider the balance between innovation and ethical accountability, ensuring that research is conducted with respect for human dignity. The discourse surrounding stem cell ethics is complex and multifaceted. Imagine a dialogue where scientists, ethicists, and the public come together to navigate the moral landscape. What are the rights of the donor? How do we ensure informed consent? These questions are vital as we chart our course through uncharted waters. Ethical frameworks must not only protect individuals, but also foster an environment where scientific inquiry can flourish, leading to breakthroughs that benefit society as a whole. Thought leaders in the field emphasize the importance of establishing robust ethical guidelines that adapt to the pace of scientific progress. This adaptability is essential in a landscape that is continually evolving, where yesterday's boundaries may become tomorrow's realities. Engaging the public in discussions about stem cell research can foster understanding and trust, allowing society to embrace the advancements that lie ahead. Envision a future where innovations in stem cell therapy are accompanied by a strong ethical foundation, ensuring that the benefits of these therapies are accessible to all, without compromising moral standards. This vision requires collaboration among scientists, ethicists, policymakers, and communities, working together to create a framework that is as dynamic as the science itself. As we look forward, the integration of technology into stem cell research will further enhance our capabilities. Imagine sophisticated bioengineering techniques that allow us to manipulate stem cells with unprecedented precision. Advances in gene editing, such as CRISPR technology, open up possibilities for correcting genetic defects at the source. The potential for creating customized cells that can target specific diseases is a beacon of hope in the fight against genetic disorders. Moreover, the future of stem cell therapy may see the rise of bioprinting technologies. Picture a printer that can create living tissues, layer by layer, using stem cells as the ink. This revolutionary approach could lead to the development of organs for transplantation, alleviating the pressing shortage of donor organs. The implications of such technology are staggering, offering the promise of not only extending life, but also enhancing the quality of life for countless individuals. The journey ahead is not without its challenges. As we refine our techniques and expand our understanding, the road may be paved with obstacles. Imagine the necessity for rigorous testing and validation to ensure that these therapies are safe and effective. The path toward regulatory approval will require careful consideration as we strive to balance innovation with patient safety. Yet the spirit of discovery is resilient. History is filled with pioneers who have pushed the boundaries of what is possible. As we stand on the shoulders of giants, we are reminded of the words of one such pioneer, who said that scientific discovery is the most important act of human imagination. This sentiment rings true as we navigate the landscape of stem cell therapy, where creativity and inquiry intertwine to forge new pathways in medicine. In this rich tapestry of future possibilities, we find ourselves inspired by the potential of stem cells to heal, regenerate, and transform lives. As we embrace the challenges and opportunities that lie ahead, the vision of a healthier future glimmers like a distant star, inviting us to reach for it with both hands. The coming years will unveil new therapies and approaches that will redefine our understanding of health and disease, forever changing the fabric of medicine. The horizon of stem cell therapy stretches ever wider, revealing opportunities that once belonged only to the realm of dreams. As we anticipate future advancements, we can envision a world where stem cell applications expand into previously uncharted territories. Imagine the potential for stem cells to play a role in regenerative therapies, addressing not only chronic diseases, but also the complexities of aging. Researchers are investigating how stem cells might rejuvenate aged tissues, promoting vitality and extending not just lifespan, but health span. The prospect of harnessing the regenerative capabilities of stem cells to combat the effects of aging is invigorating, encouraging a new paradigm in how we perceive the aging process. In the realm of chronic conditions, the development of therapies could extend to autoimmune diseases, where the immune system mistakenly attacks healthy tissues. Picture a therapeutic approach where stem cells could modulate the immune response, teaching the body to recognize its own cells as allies rather than adversaries. This innovative perspective could lead to treatments that not only alleviate symptoms, but also promote lasting remission, fundamentally altering the landscape of chronic disease management. Moreover, consider the potential for stem cells to be utilized in the realm of mental health. The exploration of neuropsychiatric disorders, such as depression and schizophrenia, could benefit from the application of stem cell therapy. Imagine a future in which stem cells can be directed to repair neural circuitry or facilitate the growth of new neurons in affected areas of the brain. This possibility could revolutionize our approach to mental health, providing hope where traditional therapies have often fallen short. As we forge ahead, the integration of personalized medicine with stem cell therapies emerges as a guiding principle. The journey toward individualized treatments is paved with the promise of genomic insights. Imagine a healthcare landscape where each patient's unique genetic profile informs the design of stem cell therapies tailored specifically for them. This could mean the difference between a one-size-fits-all approach and a bespoke treatment that considers the intricate details of a patient's biology, enhancing efficacy and minimizing adverse effects. The potential for combining stem cell research with cutting-edge technologies adds another layer of excitement. Imagine artificial intelligence algorithms analyzing vast datasets to identify patterns in stem cell behavior and patient responses. This fusion of technology and biology could lead to faster discoveries and more effective treatments. By utilizing machine learning, researchers could predict how stem cells will differentiate under various conditions, optimizing protocols and accelerating the path to clinical application. While the future appears bright, it is essential to address the ethical frameworks that will guide these advancements. As we innovate and explore, we must remain vigilant in ensuring that ethical considerations evolve alongside scientific progress. Imagine a future where ethical oversight is woven into the very fabric of research, where dialogue among scientists, ethicists, and society shapes the direction of stem cell therapies. This collaborative spirit is crucial in fostering public trust and ensuring that advancements benefit all. Consider the implications of consent in stem cell research. How do we ensure that donors are fully informed about the use of their cells? What measures can be taken to protect their rights? Engaging the public in discussions about these issues fosters transparency and accountability, allowing for a more inclusive approach to scientific inquiry. Ethical frameworks must prioritize informed consent, ensuring that individuals understand the potential risks and benefits of participating in research. Furthermore, as stem cell therapies become more accessible, we must address disparities in healthcare access. Imagine a world where advancements in stem cell therapy are not confined to affluent populations, but are available to all, regardless of socioeconomic status. This equity is vital in realizing the full potential of stem cell research, as we strive to ensure that the fruits of scientific discovery are shared widely. In the face of these challenges, the spirit of innovation persists. Scientists and researchers are continually pushing the boundaries of what is possible. Pioneers in the field emphasize the importance of maintaining a balance between exploration and ethical responsibility. They remind us that true progress is rooted in a commitment to not only advancing knowledge, but doing so with integrity. As we embrace the future of stem cell therapy, we can envision a landscape brimming with possibilities. The integration of advanced technologies, the personalization of treatments, and the establishment of robust ethical frameworks will be pivotal as we navigate this uncharted territory. The prospect of stem cells as a cornerstone of modern medicine invites us to dream of a time when chronic diseases can be managed more effectively, when the aging process can be mitigated, and when mental health can be approached with renewed vigor. The journey of stem cell therapy is one of hope, discovery, and transformation. With each step forward, we are reminded of the profound impact that stem cell research has on our understanding of life and health. As we look ahead, the promise that lies before us compels us to continue exploring, questioning, and innovating. The future is not merely a continuation of the present. It is a realm of endless possibilities, where the potential of stem cells can truly reshape the human experience. Chapter 10: Stem Cells and the Promise of Regenerative Medicine. The world of medicine has been profoundly transformed by the discovery and understanding of stem cells. These remarkable cells, with their unique ability to develop into various specialized cell types, have opened new frontiers in healing and regeneration. Imagine, if you will, a canvas where the potential for life unfolds, each brushstroke representing a different possibility, a different path toward recovery. This is the essence of stem cells. Their versatility and adaptability make them a cornerstone of regenerative medicine. Over the years, researchers and clinicians have harnessed the power of stem cells in ways once thought to be the stuff of science fiction. From treating blood cancers through hematopoietic stem cell transplants to exploring therapies for conditions like spinal cord injuries, the applications are as varied as they are promising. Picture a landscape where damaged tissues can be restored, where hope is rekindled in those battling chronic illnesses. The impact of stem cell research on medicine is indeed monumental, as it continues to shift paradigms and redefine the limits of what is possible. Looking ahead, the vision for regenerative medicine is both ambitious and inspiring. In the next decade, we can anticipate a deeper integration of stem cell therapies into clinical practice. Imagine a future where conditions that currently require lifelong management might be treated with a single intervention that repairs or regenerates damaged tissues. The potential to cure diseases rather than merely manage symptoms could fundamentally change patient care. This vision is not merely aspirational. It is grounded in ongoing research and burgeoning technologies that promise to enhance our understanding and utilization of stem cells. How might stem cells revolutionize healthcare? One can envision a world where personalized medicine becomes the norm, where treatments are tailored to the individual based on their unique cellular makeup. Researchers are already exploring the use of induced pluripotent stem cells, adult cells reprogrammed to an embryonic-like state, allowing for the generation of patient-specific cells for therapy. This advancement could lead to significant breakthroughs in treating conditions like Parkinson's disease or heart failure, where the replenishment of lost or damaged cells could restore function. Consider the stories of those who have been at the forefront of these advancements. Scientists like Shinya Yamanaka, who first developed the method for creating induced pluripotent stem cells, have paved the way for a new era in regenerative medicine. His work exemplifies the power of curiosity and perseverance in science. As he once stated, "When you find something that you love, you can work tirelessly." It is this tireless pursuit of knowledge that fuels the promise of stem cells in medicine. As we ponder the future, we must recognize the importance of ongoing research and discovery. The field of stem cell research is filled with challenges and uncertainties, yet each step forward brings us closer to realizing the full potential of these remarkable cells. The interplay between scientific inquiry and clinical application will be vital in navigating the complexities of stem cell therapies. Imagine the possibilities that lie ahead. What if we could regenerate entire organs using a patient's own cells? What if we could reverse the effects of aging by rejuvenating tissues? These questions drive researchers to push the boundaries of what is currently known, and with every experiment, new insights emerge. Each breakthrough is a reminder of the resilience of science, where perseverance and collaboration lead to discoveries that can change lives. The importance of supporting and investing in stem cell research cannot be overstated. As we stand on the brink of potentially transformative therapies, it becomes clear that the future of medicine hinges on our commitment to understanding stem cells and their capabilities. The journey of discovery is ongoing, and as we look toward the horizon, we must embrace the spirit of inquiry that has brought us this far. In this evolving landscape, the role of collaboration among scientists, clinicians, and patients becomes increasingly significant. It is through this synergy that we will unlock the potential of stem cells to address some of the most pressing health challenges. The stories of individuals who have benefited from stem cell therapies serve as powerful reminders of what is at stake, infusing urgency into the quest for knowledge and healing. As we reflect on the remarkable progress made thus far, we can also consider the importance of ethical considerations in stem cell research. Ensuring that scientific advancements are accompanied by a thoughtful examination of their implications is crucial. This careful balance will help guide the responsible application of stem cell therapies in ways that respect the dignity and rights of individuals. With a focus on the future, it is essential to imagine a world where the promise of stem cells is fully realized. The impact of these cells on medicine is just beginning to unfold, and the next decade may usher in a new era of healing and discovery. The dream of regenerative medicine, where the body itself can be empowered to heal, lies within our grasp. As we continue to explore the depths of stem cell research, it becomes evident that the possibilities are as vast as the universe itself. Each new finding invites us to envision a future where diseases that once seemed insurmountable can be effectively treated. The journey through this complex yet fascinating field is just beginning, and the potential for groundbreaking discoveries is limitless. The narrative of stem cells is not just a tale of biology. It is a story of hope, resilience, and the enduring human spirit. As we move forward, let us remain steadfast in our commitment to understanding and utilizing these extraordinary cells. The future of regenerative medicine holds the promise of renewed health and vitality for countless individuals, and it is a journey worth undertaking. What will the next chapter in stem cell research reveal? Only time will tell, but one thing is certain. The quest for knowledge and healing will continue, propelling us into a future filled with possibility and wonder. The transformative potential of stem cells extends beyond individual therapies. It encompasses a broader vision for healthcare that could redefine the very essence of medical practice. Imagine a future where the hospital is not merely a place for treatment, but a sanctuary for healing, equipped with technologies and therapies that empower the body's innate abilities to restore itself. The vision for regenerative medicine includes harnessing stem cells not just for isolated treatments, but as a holistic approach to health, emphasizing prevention, early intervention, and personalized care. As we look forward, one can envision an era where conditions like diabetes no longer require lifelong insulin therapy. Instead, scientists are exploring the possibility of regenerating insulin-producing beta cells in the pancreas through stem cell therapy. Picture the profound impact on a patient's life if they could achieve metabolic balance not through daily injections, but through a single, transformative treatment. This paradigm shift could usher in a new era of chronic disease management, where the focus is on restoring function rather than merely alleviating symptoms. Moreover, the integration of advanced technologies such as gene editing and artificial intelligence is poised to propel stem cell research even further. Imagine the synergy of these fields, where precise gene modifications can enhance stem cell function or address genetic disorders at their source. The potential to correct mutations that cause diseases before they manifest could alter the fate of generations. The prospect of combining gene therapy with stem cell applications raises intriguing questions. How might we eradicate hereditary conditions? What if we could engineer stem cells to produce therapeutic proteins that the body is unable to make? The stories of pioneers in combining these fields are already emerging. Researchers like Jennifer Doudna, who co-invented CRISPR technology, are leading the charge in exploring how gene editing can be utilized alongside stem cells. Doudna has expressed that the potential of CRISPR is vast, providing tools for precise modifications that could one day enable stem cells to be not only regenerative, but also corrective. Such innovations could redefine the parameters of therapy, enabling us to address the root causes of diseases rather than merely their symptoms. As we imagine this future, we must also consider the ethical landscape that accompanies these advancements. With great power comes great responsibility, and the potential to manipulate the building blocks of life calls for a robust framework of ethical considerations. What guidelines will govern the use of stem cells in clinical applications? How do we ensure equitable access to these groundbreaking therapies? These questions are paramount as we navigate the moral implications of rapidly advancing science. In this context, the role of public engagement becomes crucial. Scientists must communicate their discoveries and the accompanying ethical considerations with clarity and transparency, fostering an informed public that can participate in the dialogue surrounding stem cell research. Imagine a society where individuals are not only consumers of medical advancements, but active participants in shaping the future of healthcare. This inclusive approach can help build trust and ensure that the benefits of stem cell therapies are accessible to all. As we delve into the promise of stem cells, it is essential to acknowledge the role of collaboration across disciplines. The intersection of biology, engineering, and computational sciences is already yielding innovative solutions that were once thought unattainable. Bioengineers are developing scaffolds that mimic the extracellular matrix, which can support stem cells in their journey toward differentiation and integration within host tissues. Picture a symphony of disciplines, each contributing their expertise to create harmonious solutions that address complex medical challenges. The journey of stem cell research is one of constant evolution, where each discovery paves the way for the next. Imagine the excitement in laboratories around the world as researchers explore the uncharted territories of stem cell biology. Every breakthrough, whether it be a new method of differentiation or a novel application, opens doors to previously unimagined possibilities. It is this spirit of exploration and innovation that fuels the quest for knowledge and healing. In contemplating the future, the importance of training the next generation of scientists cannot be overstated. Encouraging young minds to engage in stem cell research and regenerative medicine will be essential in maintaining the momentum of discovery. Imagine classrooms filled with passionate students who see the potential of stem cells not just as a scientific curiosity, but as a means to change lives. Their enthusiasm and creativity will be the driving force behind the next wave of breakthroughs. As we reflect on the journey thus far, it is clear that the impact of stem cells on medicine is far-reaching and profound. The exploration of these remarkable cells is a testament to human ingenuity and resilience. Each step forward in understanding stem cells brings us closer to a future where regeneration and healing are not just aspirations, but realities for countless individuals. The promise of regenerative medicine is a tapestry woven with hope, discovery, and the unwavering belief in the potential of life itself. As we continue to nurture this field through research, collaboration, and ethical consideration, we will undoubtedly uncover the secrets of stem cells that can heal, restore, and transform our world. What wonders await in the unfolding chapters of this remarkable story? The answer lies in the dedication and passion of those who dare to dream and explore the uncharted territories of science and medicine.